I wanted to talk to you about DNA synthesis at JGI, which a big part of it in our mind is about converting a lot of the massive amount of sequence data that has been generated over the last 10, 15 years. And it's kind of stored in databases as digital information. And now we want to start converting that back into biology. So I'm going to break my talk into two parts. First, I'm going to give you a bit of what our vision for synthesis at JGI, some of our operation and metrics, and then after that, I'm just going to give you a flavor of the kind of products that we're doing right now that are being enabled by DNA synthesis. So the way we see things is we've been generating a ton of data from different sorts of projects, from prokaryotic isolates to eukaryotes to metagenomes, single cells. So this has all gone to feed this very large sequence databases. There's been a lot of effort at JGI also to develop very good databases that allow us to very efficiently query the databases and analyze what we find at multiple levels. But still, being able to convert all this electronic information back into applications that will help us solve everyday problems has been pretty limited. And one of the problems as we see it is that we have two very different types of information. On the one side, we have this digital information stored in databases, and what we really need is biological, biochemical molecules that we can characterize for function. And converting between these two types of information is not trivial. Some of the problems are that getting hold of template sometimes is hard, especially if you've generated your sample from a single cell or from a metagenome. But mostly, even if you could get hold of your template, the native sequences from these very exotic organisms might really not work in the model organisms that we're used to working with. Maybe the codon usage is very different. And sometimes if you're looking more than a gene, but you want to look at the function of a pathway, the regulatory elements don't transfer very well between species. And also there's a lot of cryptic regulation that we know very little about. So synthetic biology offers us a path to connect these types of information and really rewrite DNA in a way that is likely to work in our organism of choice. So that's at least the way we see synthetic biology at JGI. So our grand vision for this is to have everything as a pretty well integrated pipeline where our users come to us with interesting biological questions. These questions get addressed at JGI mostly through sequencing. So it's not only genomic sequence, it can be transcriptomics, it can be ChIP-seq, it can be the results of transposon bombings. This data needs to be analyzed through multiple levels of computational analysis, be it assembly, annotation, functional predictions, PCA, or any other type of statistical analysis that you can think that allows you to generate a number of hypotheses. And now that we have this set of hypotheses, we would like to integrate this to DNA synthesis that allows us to then start validating experimentally a lot of these hypotheses at a pretty large scale. Whatever we synthesize needs to be validated. Synthesis is a pretty error-prone process, so we need to make sure that what we synthesize is what we want to synthesize and not something that has frame shifts or other types of errors. And then we can plug our synthetic sequences into vectors that some of which we designed in collaboration with our users so they can plug in directly to the high-throughput functional screens. So once we have the vectors, this can be used for functional characterizations, either internally or they get shipped out to users, as, for example, Adam Gus presented in the previous talk. So in order to accomplish some of this vision, the DNA synthesis group was created in May 2011. Firstly, it was mostly the technological component, which I lead with Yang-Feng Chen. And lately, we've also added a more programmatic component to this, which is led by Nathan Hilson. And this is more about interfacing with the users in terms of coming up with very compelling and scientific questions, and also to deal with issues related to LC, and also pushing forward the tools for computer-aided design, which is a very critical aspect of this field. So this is what we've been doing over the last 2 years at JGI. So trying to come up with an integrated and efficient pipeline that we can productionize so that we can produce synthetic fragments at scale and at reasonable cost. So again, we always start with an electronic sequence from databases. And the first step is how do we define the strategies that we're going to use to accomplish the synthesis. And so there's been a lot of effort by people at JGI to develop, and at JBay also to develop the software to tackle the different aspects of a synthesis design. So we have, for example, this piece of software called Gene Design that will tell us if you want a gene, how do you break it into oligos, how do you code and optimize it so that it will express well in your gene, which vectors should you clone it into. Then we have J5 that allows you to go from single pieces of 1 kb synthetic pieces to much larger assemblies. If you want to go from a 1 kb piece to a 10 kb or 15 kb piece, what's the best way to do this? And then we'll also have this very unique tool called BioStudio that looks at things from a much higher level. So if you needed to design a whole genome or a whole artificial chromosome, how can you start doing that? And this is the kind of tool that will help us strategize what's the best way to do that. So once we've been thought very carefully about how we design things, we just touch the buy button, and a whole bunch of oligonucleotides get ordered. We usually work with two types of oligos, either oligos that have been synthesized on an array or oligos that have been synthesized in the classical manner. Either way, the downstream is very similar. And then we've worked out how to do this molecular biology very efficient. We have number of old generation type liquid handling robots, and we're moving from this to this new instrument, which is an acoustic printer. So instead of having to do reactions in 25 microliters, we can reduce things to 1 microliter now, and we don't have to buy expensive tips, and we can reduce the whole cost of our reagents. We generate our fragments through standard assemblies, then they get cloned, transformed, plated, and picked using the latest generations of plating and picking robots. Then our parts get pooled, and we do our sequence validation using next generation sequencing technologies. And then once we're happy that we have what we want, then this can go into a write vector, and then it can be used for the high throughput characterization. So this chart here gives a little bit of the evolution of how our pipeline has progressed over the last couple of years. So the red line here shows you our output per quarter in terms of sequence verified 1 kb fragments, and in the last quarter we did about 700 kb of synthetic DNA, and we're hoping to reach about a megabase per quarter in the next quarter. And the blue line shows our cycle time, and again this is for the short 1 kb fragments, and now it takes us about 2 weeks to process a batch of 100 to 200 1 kb fragments. Of course, this is not necessarily what our users need. Our users often demand things that are 10 or 20 kb in size, and that takes extra cycles of our pipeline, and that's a bit longer time. So this is what I was referring to in terms of what our users want. So when we started this, most of our users wanted gene-sized fragments. This is the blue chart here, 1 to 1.5. So most of what we're doing was building these short fragments, but as time passes, we see that this has changed pretty dramatically, and in the last quarter of last year, for example, most of what we did was large fragments of 5 kb and larger. So in this area of building large fragments and combinatorial libraries is where I think we're going to put a lot of our efforts, and we can carve out a niche. So our capacity for this year is 3.3 million bases of synthetic DNA. And this year it was allocated mostly to our BRC collaborators. There's a fragment that went to the CSP program that I'm sure you're all familiar with. And some of the capacity also went to internal research projects. So we've been synthesizing things for about 2 years, and next I want to give you a flavor of the type of projects that we've been using, that we've been performing. So starting at the top here is an example of a project where we're analyzing single genes, 1 gene at a time. And so the goal of this project was to do a very broad phylogenetic scan of the diversity for a single family of enzymes called Gh1s. And the point was to try and look whether in all this phylogenetic diversity that exists in nature, we can identify enzymes that will be very robust in an industrial setting. We then have been moving on to projects where instead of looking at one gene at a time, we're now looking at small operons. And this particular example that I'm showing here concerns this synthetic carboxysomes. So carboxysomes are small organelles that are involved in carbon fixation in bacteria. And this is a collaboration with Cheryl Kerfeld, and also started as a CSP project. So what we've done is, so typically, to have a functional carboxysome, you need at least 5 to 6 genes, and they tend to occur in clusters. And what we've done is we've synthesized orthologs for each of the genes in this pathway from a diverse set of cyanobacteria, and they've been combined randomly. So we've created a combinatorial library of synthetic carboxysomes, and in a single experiment, we were able to generate about 40,000 combinations that can now be tested for function. So this is an interesting example of the power of synthetic biology. The next project is different to all of the rest because it involves the creation of a versatile and safe chassis for synthetic biology. There's not a lot of synthesis as such in this program, but we're trying to do targeted modifications of the genome at a very large scale. And I'll talk about this a bit later. And all the way to building a whole artificial chromosome in yeast. So as you see, we go from single genes to operons to genome-wide targeted modifications to building a very large synthetic construct. So the two projects I want to focus on now are this Gh1 project and the chassis. So the Gh1 project started as a collaboration with Blake Simmons, and you're all aware that one of the limitations for the development of cellulosic biofuel is the lack of sufficiently active and robust enzymes for the biomass breakdown. And at the time, they were interested in one particular family of cellulases, this Gh1 family that break down cellobiose into glucose. And they weren't looking for just any enzyme, but they wanted an enzyme that worked on very specific conditions. So they wanted something that would have good activity at 70 degrees, that would be stable at a pH of 4.5, and most importantly, that it would be resistant to ionic liquids. So this is ionic liquids is what they use as their pretreatment method. So our approach for this project was to see what's in there in nature and try and really sample the wide phylogenetic diversity for this gene. So what we did is we went into databases and tried to pull out everything that looked like a Gh1 from databases. And also at the same time, we had recently completed the cow metagenome project, the cow rumen metagenome project. And so we looked at that data and were able to identify about 50 additional Gh1s that were present in the cow rumen. Everything got aligned, and we built a tree. And as you can see, we were able to identify Gh1 enzymes from all kingdoms of life. So everything labeled in red comes from bacteria, green from archaea, and blue from eukaryotes. And this purple stripes here are the ones from the cow rumen. So one thing immediately that was very apparent that even though the cow rumen genes were only 50 from the whole set of 1,000, they really contributed to expanding the phylogenetic diversity in our tree. So for example, if you look up here, there's a branch that is uniquely defined by the cow rumen genes in this very interesting area of the tree. And we find this again and again, so these branches are defined only by the cow rumen genes. And I think this is a theme that will be quite recurrent, that if we want to expand the repertoire of protein diversity, I think metagenomes are really going to help us with that. So anyway, we have this tree, and we wanted to sample it from the tree in an unbiased way, so as to try and capture as much phylogenetic diversity as possible. And this is this red dots in the outer part of the circle. So these were the genes that we went ahead and synthesized. And you can see that some part of the tree where the branch lengths are pretty long were very heavily sampled relative to other areas of the tree around here, for example, where just one representative, because the branch lengths are pretty short here. So just one representative is enough to capture most of the sequence diversity in that area of the tree. So we wanted to synthesize this 200 genes. We were able to synthesize about 180 of them. They were all put into expression vectors, and they were overexpressed in E. coli. And we found that about 65% of them were, we saw a band of 50 kilodaltons showing that this proteins folded correctly and were solubly expressed. Then we started characterizing them in terms of their function. So this is I'm showing here enzyme activity at different temperatures. So we see, for example, this enzyme here, 159, it works best at 50 degrees Celsius, but after you raise the temperature a little bit, the activity completely drops. But we see other examples of enzymes, like this enzyme 22, where we see very high activity levels going all the way from 30 degrees all the way to 90 degrees. So this is the kind of enzymes that we were looking for. However, our study design called for all of our 180 genes being profiled at 4 different temperatures, at 2 different pHs, and against 3 different substrates, and everything needed to be done in 3 replicates. So that's a total of 15,000 reactions. And of course, we didn't want to do this in the standard colorimetric plate-based assays because it was going to take us a long time, so we needed a high-throughput method to do this. And we were lucky that at the same time, Trent was ready to really start using the methods that he had developed, NIMS, at quite large scale. So this NIMS method is a nanostructure initiator mass spectrometry. So basically, you can spot your enzyme and substrate reactions on a solid array, and very quickly, each of these spots gets hit by a laser. The reaction gets ionized and generates spectra that one can use to quantify the conversion of substrate to product in a semi-quantitative way. So this is what the NIMS data looked for us. It's an example of that, and every spot here corresponds to a single enzyme substrate reaction, and the color represents the amount of conversion that we observed. So these white spots mean that the substrate was converted almost 100%, and the dark areas means that there was very little conversion, thus inactive enzyme. And this is what the data looks if you go and look at the mass spectra. So here, for example, you see the Cellobios product, substrate, and the Cellobios product indicating there was quite a lot of conversion. On the other hand, we can see maltose. We can see a peak for the substrate, but no peak for the product, suggesting that this enzyme was not active against maltose. So if we summarize all 15,000 data points, we can do that in a single tree. So this is the tree now of the 200 representatives that were sequenced. And we have 4 data tracks. The first track is the expression, so everything that is black means that was solubly expressed. And then we have activity against Cellobios, Lactobios, and Xylobios. And the color code represents the maximum temperature at which we observed activity. So for example, this enzyme here was active against Cellobios and Lactobios, and it was active all the way up to 90 degrees, whereas enzymes around here were only active at 40 degrees and not higher. So we immediately saw some very interesting patterns from the data in this tree. For example, although we saw a lot of the thermophilic enzymes where we would expect them to be near the archaeal branches, we also saw other areas of thermostable enzymes in other parts of the tree. Some of them correspond to environments where you expect to see thermostable enzymes, but you also like to see areas around here where these are mesophiles, and they also have enzymes that have thermostable properties. Then the next thing if you remember what JB was looking for were enzymes that were thermostable, but also that they were resistant to ionic liquids. So that's the next thing we tested. So we took all of our enzymes that were thermostable, and we tested them for tolerance against ionic liquids. We run assays at 70 degrees Celsius and 20% ionic liquids. And we did this for 10 minutes. So this is the blue bars, and we see that most of the enzymes have at least some activity under these conditions. But this is not really relevant to any industrial setting. Usually, when you do an industrial fermentation, you want things to go on for 2 or 3 days. So we need enzymes that keep this activity at these conditions over long periods of time. So what we did was a pre-incubation of the enzymes even before adding any substrate for 24 hours. And then we measured the residual activity of the enzymes after the 24-hour incubation. And we see that at least 5 of the enzymes still have a substantial amount of activity. And they map to different parts of the tree, and in some cases they are not exactly where you would expect them to be. So it just shows that this metagenomic based approach of widely sampling the phylogenetic diversity is really useful. It's not, so you don't only see what you expect to see, but you get a lot more information that you necessarily expect. So from this product, we synthesized 180 of these Gh1 proteins. They were characterized in terms of their expression, activity at different temperatures, and resistance to ionic liquids. No previous enzymes were really known to work under the conditions that we were given. And after performing this project, we identified at least 5 enzymes that had significant activity after 24 hours of incubation at 70 degrees and 20% IL. And our partners at JBay are further studying the potential use of these enzymes for more industrial type applications. The second project that I wanted to talk about was this construction of a safe and versatile chassis for Symbiote products. So for this project, our definition of a chassis is a bacterial system that has been engineered with features that enable it to serve as a safe and convenient platform for us to be able to do subsequent synthetic biology experiments. So the features that we're looking for is that we want it to be very safe. We don't want the DNA to be transferred easily to other organisms. We want it to be robust, so resistant to phage infections. And we want its growth to be restricted to the laboratory. And our starting point for developing a chassis was this Corynebacterium glutamicum, and we chose this bug because of its relatively small genome size, its fast generation time, metabolism is quite well understood, it's easily transformable, so you need something that you can work with. It's biosafety 1, and it doesn't really pose a threat to humans, and especially because it's a very industrially robust organism, so it's currently used for the industrial production of amino acids. So the first steps for us in order to convert wild-type glute into a chassis is to develop a codon-depleted strain of glute. And in our case, we wanted to remove every instance of the TGA stop codon. We then needed to remove the machinery that interacts with this stop codon. In this case, is this release factor 2. And then eventually, we can also add machinery that translates the depleted codon into something else. So it's an orthogonal translational machinery. So this is what the wild-type glute looks like. So it has 484 opal stop codons. And what we want to do is convert each instance of this stop codons, TGAs, into a TAA. And as you see, the TGAs were associated with this release factor 2. So now we can get rid of this release factor 2, and it shouldn't affect very much the function of the TAA codon, as release factor 1 is also involved in TAA recognition. So this will generate a glute 1.1 strain that will not have any stop codons, any TGA stop codons, and no release factor 2. And this work is currently in progress. So what properties would we expect this codon-depleted strain to have? So first, we would expect it to be theoretically resistant to phages. So phage infect most bacteria, and they get their proteins transcribed and translated by the protein. Most of the phages that we know of in C. glutamicum have multiple opal stop codons in essential genes. So in the wild-type situation, the phage will be able to translate and transcribe and translate its proteins and complete its life cycle. But in a codon-depleted strain, in which we've also removed the release factor 2, every time that a ribosome sees an opal stop codon, it will stall, and these proteins will be tagged for degradation. So the hypothesis is that the phage will no longer be able to complete its life cycle successfully. The second property that we're interested in was transcript security, and for this we required, again, the codon-depleted strain, the RF2 knockout, and in addition, we require also an orthogonal translation machine, so we need a tRNA or a tRNA synthase that instead of recognizing TGA as a stop, will recognize it as a different amino acid. So in this case, for example, an arginine. And why this is interesting is that because then with every synthetic construct that we make in this strain, we could include opals that will be interpreted as arginines. And because in our strain, we have this orthogonal translation machinery, this proteins will be correctly translated. But if, for any reason, our synthetic genes were to leave our organism and go to other organisms through horizontal gene transfer events, they will not be correctly interpreted. So every time another cell sees this opal, it will interpret it as a stop codon, and thus the synthetic proteins will not be correctly translated in other organisms. So it adds a level of safety and control. The third property that we're looking for was this biocontainment property. And for this, on top of the properties of the engineering steps that I mentioned before, we want to make this orthogonal machinery inducible. And we want to pepper essential genes in the C glute genome with opals. So that in the lab, if you provide the inducer, the essential genes will be correctly translated and transcribed because you're also inducing the orthogonal translational machinery. But if the organism were to escape, in the absence of the inducer, a lot of the essential genes, for example, polymerases or translational machinery, will not be made, and the organism should not be able to survive. So this is a work in progress. It's an ambitious and challenging project, and the actual biology of this strain, we can make theoretical predictions, but we'll have to see what actually happens. But a very good side effect of this is that we're developing new technologies for doing this targeted genome replacements at quite high scale. And this is potentially a very new and exciting capability that perhaps in a couple of years' time we will be able to provide our users with. So to summarize what I've told you before, we really have a vision for growing this synthesis program at JGI. But for it to be successful and have an impact, I think we need to comply with a number of critical steps. So we need to have an efficient synthesis pipeline that will generate things like short fragments, long fragments, combinatorial libraries, and also targeted genome modifications. We need to be doing very compelling science. I think we're fortunate to be in the Bay Area, and there's a lot of activity in synthetic biology, but we're also counting on close interactions with our BRC collaborators, and also with our user community coming up with very interesting and exciting projects. We need to be able to do this in a safe and reliable manner. That's why we're working on the development of the chassis. If there's a public perception that this is very dangerous, I think we will not really be able to do it. And then I cannot emphasize enough the importance of having computer-aided design tools that help us do this. In a way, I think that we're putting a lot of effort developing synthesis technology, but whatever we do, synthesis is going to become a commodity in a few years' time. But I think knowing how to build a construct that will actually work will be very difficult to commoditize. And that's why having this know-how and these computer tools that are pretty unique will keep us very competitive. Last thing I want to say is that, as I said before, we're counting on our users to come up with an interesting science program. And on April 8th, I think, is the deadline for our users to submit letters of intent for sequencing as well as for synthesis. And in particular, for synthesis, we'll look at every project that we get. But what we would really like to encourage are projects that address refactoring, screening, and functional characterization of multi-gene pathways that are involved in DOE mission-relevant areas. We like things that involve large molecules. We don't want all of our molecules to be large because it's going to make our lives harder, but having a substantial amount of what we synthesize being large would be good. And things that could be coupled to high-throughput screening, I think, are also great. So we're not exactly sure how much we will allocate to CC projects next year, but I think around 2 megabases is about a good guesstimate. This should allow us to do about 5 to 8 projects, and the typical allocation for each of the projects is about 250 kilobases per project. So with that, I'll be happy to take any questions.
Question in the back.
Hey, Sam. So what sort of informatic infrastructure are you guys putting in place to sort of take all of the data that's going to be generated from this and be able to use it to make predictive or build predictive knowledge about the chassis or the organism that you're working with, and hopefully expand that out to understanding other organisms? Because that seems like the real rich source for synthetic biology.
Yeah, no, I completely agree with you, and it's something that yeah, we want to develop with our partners at K-Base. It's not something that we've developed already, but it's something that we're thinking about.
So how much does the cost per kilobasis?
So this changes all the time. Right now, we just did a cost calculation last week, and it's about 17 cents per base pair. But we hope this will go down every quarter, and we're working quite hard to make this happen.
Is your major cost oligos, the labor associated with putting the pieces together, the design?
Yeah, about 60% is cost, is oligos. Maybe 20% labor, and then overheads. And
other questions for Sam?
It seems to be an interesting convergence of the opal codon story today from Tonia's work and yours. You guys worried about transfer to SR1 or something?
Right, yeah, that's a good point.